DNA experiments have total 7 steps. Clear?
Yes.
First is sample selection, sample homogenization, cell lysis, protein precipitation, then DNA precipitation, DNA washing, storage. Right? After that, we do the electrophoresis. In electrophoresis, first the gel preparation, 0.8 gram agarose powder in 100 ml buffer solution. Boil it. Before it cool, we add ethidium bromide in the dye. Ethidium bromide attached to the DNA when it run in electric current, and it gives the color, orange fire color, glow up. That's we record through the camera and mobile camera. That's become the evidence, records of DNA profile, DNA bands, genomic DNA purity, intactness, everything's recorded through that gel electrophoresis, right? In first step, sample selection, you will follow 5 points. Sample should be soft and new, easy to handle, easy to grind, easy to cell lysis, and free from contamination. Then come to the homogenization process. In the homogenization process, what you will do is sound-free grinding, stroke-free grinding. No sound, no stroke. Sound generate energy that can break to the DNA. जैसे बम ब्लास्ट से खिड़कियाँ टूट जाती हैं, ठीक? तो हम साउंड फ्री ग्राइंडिंग करेंगे, टक टक की आवाज़ नहीं करनी है. दूसरा, लो टेम्परेचर पर हमको ग्राइंड करना होता है. लिक्विड नाइट्रोजन साइंटिस्ट यूज़ करते हैं सैंपल की ग्राइंडिंग में. लिक्विड नाइट्रोजन -196 डिग्री टेम्परेचर, राइट? सो हम क्या करेंगे, मोटर पेस्टल को फ्रीज कर देंगे फ्रीज़र में, ऊपर वाले फ्रीज के पार्ट में, जिससे वो चिल्ड हो जाएगा. उसमें ग्राइंडिंग करते हैं अगर हमें और कूलिंग की ज़रूरत महसूस होती है तो. ठीक? क्लियर? इसके बाद सेल लाइसिस आता है. सेल लाइसिस इज इक्वल टू द मसाला फ्राई इन किचन. एवरी साइंटिस्ट यूज देयर ओन recipe, their own composition. Like every cooks utilize different composition of spices. Same way here will be change. Everybody do some change in chemically, but 5-6 same chemical used by everybody, right? Then come to the protein precipitation, DNA precipitation, DNA washing, DNA storage. These 4 steps are the common steps. So all follow the same step. Any plants, any sample, any whatever, you follow the same steps, right? That's called common protocol. In common protocol, there is common solution. Name of solution, like protein precipitation, do the protein precipitation, DNA precipitation, do the DNA precipitation, and DNA washing, do the DNA washing, DNA storage, do the DNA storage. First chemical that you use to in DNA isolation process is trace. Fix the pH of solution. EDTA bind Ca++ Mg++ ions to stop the DNase RNase enzyme. Then NaCl maintain ionic strength. Either CTAB, either SDS you will use that to selectively utilize for animal world and plant words DNA experiment. If you use SDS, you can extract DNA from animal words. Any heart, liver, kidney DNA, mosquito DNA, earthworm DNA, you can do that. If you utilize CTAB, then any fungus, bacteria, algae DNA, you can do with that CTAB molecule, right? That is the basic fundamental principle. After DNA photography is done, after PCR done, then you do the electrophoresis, photographs of DNA is come, then second part is start, that's called dry lab. Dry lab means analysis work. Analysis work is called dry lab. And those experiments you did today, it's called wet lab experiment. Means जो हम experiment करके data generate करते हैं, that is wet lab experiment, real lab experiment. After that analysis work start, like X-ray image. You take the X-ray image, but you can't understand. Doctor explain it is what? After analysis of that, so somebody know only the wet lab work, somebody know the dry lab work. Those who know both the work like professor. Those only know how to extract the DNA, but does not know how to analyze it, how to find it, where is gene, where is allele, what is pattern, what is diagnostic marker, what is permanent marker, what is there, all things those are does not know, they are work like labor. But if you want to work like scientist, work like professor, work like as analyst, work like as a study more deeper, you must to know the analysis of DNA science, DNA bands, DNA profiles, patterns, and its uniqueness. Right? So that part we will cover through the lecture and finish this total chapter, including isolation, electrophoresis, DNA photography, plus DNA fingerprinting, molecular marker analysis, DNA diagnostics, right? DNA diversity analysis, pattern analysis, profile analysis, parental analysis, ancestral analysis. That's all come through the lecture, right? Thank you very much, everybody.